Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Marc Vermulst. He's an assistant professor of gerontology at University of Southern California. We're going to talk about aging. So, Marc, thanks for coming.
Sure. Thank you.
Yeah, how did you get interested in aging? When, was it a long time ago, and what got you interested?
Well, it wasn't actually a very long time ago. I remember that I was 8 or 9 or something, and I remember lying in bed and asking my mom what happens after we die. My mom told me a really fun story about how people had come back from the dead and it was a wonderful place. And I think I believed that for about 30 seconds or something. And then I realized, oh, wait a minute, she's just telling me something to make me feel better.
Right, yeah.
So it was really this sort of, existential angst when I was a tiny kid. But as I grew up, I stayed interested in it, and really my goal is to understand my own fate. So it is my personal fate to grow old and die, and I guess it's not just mine; it's also yours. Everybody listening to the podcast, and not just everybody who's living now, everybody who's lived in the past and will live in the future. Every organism on the planet is fated to grow old and ultimately die. So really the goal of my research is to understand the fate of every living organism on the planet.
So what aspects of the aging process are you studying in particular then?
So I am particularly interested in mutagenesis. So as we grow older, our genome changes. My specific skill is that I'm very good at being able to measure how quickly our genome changes and how that impacts our health. And that really started when I was somewhere in my 20s when I learned about so-called premature aging syndromes. These are patients that have a disease by which they age at an accelerated pace.
Like progeria and stuff like that?
Yes, right. There are a whole bunch of them, but something they have in common is that most of them are characterized by an instable genome. So their genome changes at a faster pace than ours. And that got me interested in trying to understand the relationship between mutations, the repair of our genome, and the aging process.
Well, when you say the genome changes, you mean epigenetically, or do actual base pairs rearrange, and in which cells? What
right.
changes?
The actual base pairs. So every time a cell divides, it needs to copy its entire genome because the daughter cell needs the exact same instructions. And our genome is huge, right? It is 3 billion bases. So copying 3 billion bases perfectly is extremely hard. And every cell division, approximately 60 to 100 changes happen. And that can have ramifications that are not apparent until much later in life.
Has anyone done a survey of all the cell types to see the aging speed of all of them and the rate of change and the degree of change, and put that on a chart for analysis?
There are certainly charts like that. It depends on many different factors. For example, certain cells, for example, in our gut, right? Our gut renews itself completely every 3 days. So there are completely new cells lining the inside of our gut every 3 days. And because they divide so much, they accumulate these changes faster. In contrast, cells like neurons or muscle cells or heart cells, they don't really divide ever. So some neurons are born and they stay with us for 80 or 90 years. And during that time span, they don't divide even once. So in those cells, it's much slower. But there are other reasons why those cells might age.
So which cell types are you studying in particular, and what do you see?
So I study mainly neurons and cells of the brain. I am particularly interested in aging of neurons because I care about Alzheimer's disease and Parkinson's disease and all these types of age-related neurodegenerative disorders. And what we focus on there is not necessarily the rate at which the genome changes, but the rate with which changes occur in the messages that are made from our genes. So whenever a gene is activated, a temporary copy is made, and the temporary copy is shipped out to a different part of the cell to make another protein. And as it turns out, these messages, these temporary copies, contain errors as well. And it seems as though, from the work that we've been doing, that those could be a really important part of the origin of Alzheimer's and Parkinson's disease.
So what changes? Is it just individual base pairs, or is it large regions that transpose or disappear or come in?
They are mainly single base pair changes. And the problem is that as a result of those changes, the protein that is made from that temporary copy does not fold in the right conformation. So it is partially unstructured often where the error happened. And you may or may not know that misfolded or badly constructed proteins are at the heart of all of these neurodegenerative disorders. So what we do, for example, is we generate cells that make more of these errors than normal. And then we study how that impacts various aspects of Alzheimer's. And Alzheimer's disease is caused by misfolded proteins, at least partially. And what we find is that in cells that make more of these errors in these temporary copies, that process is accelerated. And we find that these types of errors occur more frequently in cells that are older compared to cells that are younger. So one of the reasons that we think
what makes a cell older? Is it the number of divisions it's gone through? Like, is it near the Hayflick limit, or is it that the time between the last division was longer?
I guess in this particular case, in the case of neurons, it would be a whole range of different aspects. It would be their ability to generate energy. It would be potentially epigenetic changes. It would be the accumulation of macromolecular damage. It would be the production of toxic side products, such as reactive oxygen species. There are a whole range of different things that come together in order to age cells. And one of the consequences that we found is that as a result of all those different things impacting the aging process of these cells, they start to make more of these errors in the temporary copies, and that ultimately contributes to Alzheimer's disease. So we are trying to link the natural aging process, link that in a mechanistic way to age-related diseases. Because one of the kind of funny things is that everybody knows that diseases like Alzheimer's and Parkinson's are age-related diseases, right? But what it exactly is about the aging process that causes those diseases is not clear. And so we hope that we have found one of the mechanisms by which natural aging ultimately contributes to the development of these age-related disorders.
So when you look at the cells, you're looking at the neurons, you're looking at them across, people that are of various ages or people that have disease or don't have disease. Like, what's your cell population come from?
So we mainly work in cell models. So we work on yeast and worms and flies and mice. And we work on embryonic cells that can potentially be transformed into neurons. And we're doing experiments specifically now, for example, on brain tissue of young versus old mice. And we are basically probing all these temporary copies for errors. That's our main focus now.
So do you see it's a threshold of a number of misfolded proteins that causes the disease, or is it just one? Like, what do you observe about it?
Well, so that is one of the most important things, probably. One of the most exciting things I personally think that has come out of the last 20 years of research is that a lot of the neurodegenerative disorders, including Huntington's disease, Alzheimer's, Parkinson's, amyotrophic lateral sclerosis, a lot of those diseases seem to be caused by proteins that develop a life of their own. They become what is called prion-like. Prions are special proteins that can propagate themselves. They fold in a funny way, and as a result, they can force other proteins to assume their conformation. So if you have one of these prion-like proteins, it can force other proteins to assume its shape, and they then aggregate in these really strong, structured lattices that seem to be toxic for the cell. So it's self.
Well, what do you mean force? You mean they just provide like nucleation sites for proteins to fold in such a way as they conform to that?
Yes, exactly.
That nucleation.
Yes, exactly. So one of the things we're very interested in is the possibility that these temporary copies, these mRNA molecules, that errors in those copies accidentally give rise to proteins that now have prion-like properties, and as a result, this autocatalytic cascade is started that ultimately results many years later in a neurodegenerative disorder. One of the things that has puzzled me for a very long time is that when we're looking for the root cause of a particular disease, what we tend to do is we tend to focus on what is wrong with our genome. Is there a mutation that can explain why we get Alzheimer's or Parkinson's or some other disease? Sometimes that is true. Sometimes you can find a mutation in the tau gene or in a pre-senilin gene that explains why a person gets Alzheimer's disease. But in most cases, the disease occurs sporadically. I think the latest estimate is that about 10% of cases of Alzheimer's disease are caused by genetic mutations in the germline of the patient, and 80 to 90% are sporadic cases, meaning we don't know what the really cause by. And there have been a lot of.
If you look at the underlying, are these SNPs? Is that what these alterations are? These mutations are?
Well, I think it's probably not correct to call them a SNP. A SNP would be a mutation that is extremely frequent in the population, and these mutations are rarer. So I think I would simply call them an inherited mutation, probably one every 10,000 to 100,000 people. Whereas SNPs are.
I mean, the 80 to 90% of people that get Alzheimer's, for instance, what's the correlation there? Are there certain base pairs that tend to be modified, or is it appearing to be random, or what?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Right. No, so that is the problem. If there's no genetic cause, what is the reason that these people get Alzheimer's disease regardless? And so one of the things that we're thinking about is that the change that caused the disease is not present in the genome, it's only present in these temporary copies. And that is why it has been missed up to this point, because these temporary copies are not normally surveyed for mistakes. So we think, we hope.
Why would there be an error in a copy? Is this an RNA issue then?
Yes.
Maybe there's a genetic change in the RNA that doesn't. So the transcriptome is modified.
Yes, exactly. So we're thinking about it in terms of a perfectly normal gene that would normally give rise to the correct RNA molecule and the correct protein, but something goes wrong during the transcription process, and as a result, the RNA polymerase that is making the transcript makes an error for whatever reason, and that results in the wrong protein that could have potentially prion-like properties. So we're thinking that these temporary errors, these errors in these RNA molecules made by RNA polymerase II, that those are ultimately a really important source for the generation of these prion-like proteins. Because as soon as you make a few of them, they will self-amplify in this autocatalytic cascade. And
if you trace back the origin of these modified RNAs, where are they coming from? At some point, they're coming from either the mitochondrial DNA or maybe the ribosome or the nucleus itself, right?
Right. So there are probably a lot of different reasons why those errors happen, and we're extremely interested in those mechanisms, because any mechanism that would increase the error rate of transcription could potentially accelerate the age of onset, the progression, and the severity of neurodegenerative diseases. So we have been looking partially at DNA damage. So if a gene is damaged, it is more likely that an RNA polymerase will make an error on it. Normally, these errors occur once every 100,000 bases. But if there is damage present, that rockets up to 50%. So every 2 transcription events, an error will be made on one of these damaged genes. So one of the reasons why we're particularly interested in that is because there are well-known cases of pesticides and herbicides that cause DNA damage, and farmers that are exposed to these DNA damaging compounds have an increased chance of getting Alzheimer's or Parkinson's disease. So we think there is additional data there for a link between these transcription errors and neurodegenerative diseases. And the other thing that you said is probably also true. As our cells age, particularly cells that are highly metabolically active, as are our neurons, you might already know that our brain is only 2% of our body weight, but it uses 20% of the oxygen we consume. As these neurons age, the mitochondria become more dysfunctional and they spew out more reactive oxygen species. And those are really strong catalysts for making these transcription errors. So we're thinking there are really strong links between DNA damage, either through exogenous sources like things we eat or drink or we're exposed to, or endogenous sources like the natural aging process.
Has anyone looked at is the prions, once they act as nucleation sites, is that what creates the plaques that form?
Yes, that seems to be the case. There are many, many ways in which these plaques can form. They can have many different structures, but especially in the last 2 to 3 years, multiple papers have been published that show that it is really these specific amyloid proteins, these specific nucleated aggregates that have infectious properties, so prion-like properties, that are the primary drivers of the disease. These prion-like proteins can be generated by genetic changes. So, for example, patients with mutations in particular locations of these aggregation-prone proteins show increased prionogenic potential. And the exact same changes that are present in patients with these congenital or inherited mutations can also be generated by transcript errors. And the difference here is that genetic changes are extremely rare, but these transcript errors are relatively common. So there are approximately 100 to 1,000-fold more transcript errors made than genetic changes. So when I think about a brain and I think about the fact that there are 85 billion neurons in it, that are constantly churning out transcripts that are used to generate proteins required for brain function, some of which have the potential to be prionogenic, but need this extra little push, right? This extra little push being a transcription error or a genetic change. I personally think that every single day, countless transcription errors give rise to these prionogenic proteins or proteins with increased prionogenic potential. And that is ultimately the source of the proteins that result in Alzheimer's or Parkinson's disease when we're older.
Yeah, but there's got to be some cleanup mechanism that denatures the prions or makes them inert or something to prevent these plaques. Otherwise, we'd all be,
Yes, exactly.
quick.
So what needs to happen is that for whatever reason, these prionogenic proteins or these plaques that are forming, these small aggregates, they need to be able to evade the protein quality control machinery. There is an enormous amount of machinery present in our cells that is solely focused on degrading misfolded or dysfunctional proteins. I'm thinking about heat shock proteins, for example, molecular chaperones that refold proteins that did not achieve the right conformation. Secondly, there is autophagy, which is a more decisive mechanism that simply degrades proteins that cannot be refolded, and that is complemented by the proteasome, for example. Proteins that not fold properly frequently get tagged and are sent to this meat grinder of some sorts to be degraded. So the prionogenic proteins that are formed, they won't immediately cause the disease because they will be degraded relatively efficiently. But as we grow older, all these cleaning crews go on strike and they function less efficiently. And at some point, there is a tipping point where there are more toxic proteins produced than are degraded, and that allows them to see these aggregates and nucleate into these harmful plaques.
Well, very good. It's just about out of time. But what do you see as an hopefully in the next couple of years? I know this is a field that it's been very difficult to figure out, but do you feel like you're on the trail of, I don't know, some kind of breakthrough in the near future?
Thank you. I couldn't hear completely what you said. I heard the last bit, but this is what we're really hoping for. We're hoping to have found a novel component of the etiology of all of these different diseases, and we're trying to completely change the rule book on what the origins of human disease are. Right? We want to add a completely novel component to it, and that is the long-term goal, to discover something new, something medically relevant, and something we can hopefully act on in the future.
Well, very good. What's the best way for people to find out more about your work?
I think that my work is mainly published in scientific magazines, but if people are interested, they can look up my website at USC. And we have many links there to additional research avenues. And if people are interested in joining my lab as a graduate student or a postdoc or as an undergraduate researcher, they are more than welcome to apply. We're always looking for excited young scientists to push our research further.
Well, very good, Marc. Thank you for coming, and your research is very, very important. I appreciate you being on the podcast.
Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.